Anyhow, I'm in the product management team, and I'm responsible for the bioinformatics product at Oxford Nanopore. So over the next few slides, I'm going to try and explain what our product is, where our product came from, and what we would like to see our product evolve into in the coming weeks, months, and years. So why Oxford Nanopore? Well, this is the biological pore protein. The biological pore is a fundamental part of bacterial systems. It's a way that bacteria communicate with each other. It's a small hole that transports information, often plasmids, often other pieces of DNA. Going back a few years, there was a concept. Could we use the biological pore protein as a way for sensing particular biomolecules as they transit the pore? And the answer was yes. This is the center of, or this is the concept of nanopore sequencing. So the animated graphic behind is giving just a brief overview as to how nanopore sequencing really works. So you can see the DNA molecule. The DNA molecule has been recruited to the nanopore, and there is the motor protein is feeding single-stranded DNA through the pore. The pore protein has been genetically modified to our specifications to give particular characteristics through which we can then observe the flow of current across the membrane on which the pore has been localized. The DNA transits the pore at a speed of about 400 nucleotides per second. We have multiple pores on a synthetic membrane, and we can obtain information from each of these pores independently. As soon as the DNA has transited the membrane through the pore, then we have a trace of signal, the picoamp flow over time, and this is immediately available to be base called. And with the base called information, we have a DNA fragment against which we can start performing bioinformatics analyses. So we're a DNA sequencing company. We have a number of shiny machines that enable the sequencing of DNA. They range from the small, the MinION, smaller than a cell phone, and they range to things the size of, how big's a PromethION? I'm not quite sure the metaphor I meant to use to describe it. Anyhow, it's the size of a small microwave, a compact microwave, a flattened microwave. Anyhow, these machines give an ability to go and sequence at very different depths of information. And particularly the smaller machines enable a democratized sequencing where you can take the device out into the field. You can take your DNA sequencer to your biological sample rather than bringing sample to your sequencer. So how does this work? Well, the machine travels to all sorts of interesting places. On the left, we've got sequencing in space. Yes, you can sequence extraterrestrial DNA. Okay, the DNA is probably of terrestrial origin, but it was sequenced in space, thus it is extraterrestrial DNA sequencing. Perhaps more attractive to some of us is sequencing on the Indian Ocean. Perhaps we want to sequence in a submarine. More or less, you can take your sequencer wherever you want, and this is the democratization of DNA sequencing. Let's sequence in the environment that makes most sense to your application. Now, this generates an interesting question. If we're truly going to democratize DNA sequencing, then should we also not democratize the bioinformatics analysis? What's the point in generating data in an exotic climate, in an exotic setting, if you then need to store it on the memory stick, you need to bring it back home, you need to go and load it into your data center, and you need to wait for the data analysis? So can we do more? Well, let's start having a look at what is Oxford Nanopore offering in terms of bioinformatics analysis. Traditionally, we talk about three different pillars through which we can empower analysis. On the left, we have our code that is deposited in GitHub. We typically call these as research releases. They are for the brave. They are for you folks, the developers. It requires an understanding of makefiles. It understands the ability to install software through PyPy. It's for the brave. And if we're democratizing bioinformatics analysis, we need to empower those who are perhaps a little less brave when it comes to bioinformatics. On the right, we have Epitome. This is our cloud solution through which we can empower, a fairly simplified, robust computing in the cloud. Data is uploaded, the analysis happens, a digested report comes back, and all of the important files are written to your disk. So this is really, the simplification as to what we should be working towards. And then in the middle, we have what we call Epitome Labs. And this is a little bit of a playground. It's a formal product. It's where we're beginning to see what we can do with Nextflow. And this is what we're going to be talking about over the next few minutes. So, what was the original brief for a non-cloud-based bioinformatics? Well, the original brief was we needed something that could be run by our users, and it needed to run on our GridION device. Our GridION device is a friendly sequencing machine. It has perhaps more compute capacity relative to the amount of sequence that it can generate to others, and this is a machine that is widely found in core facilities. Core facilities, as we've discovered, have all sorts of projects to go on, so we have a platform here where we want to enable our users to start running slightly more ambitious bioinformatics workflows. How can we help our users help their users? How can we help the core facilities resolve the problems? So yes, can we implement something on the GridION device? And by extension, can we also enable other users, particularly MinION users, to use their own computers to perform bioinformatics analyses? So, in the beginning, there was the wish, and our solution was to use a Jupyter Lab environment. This was Dockerized. We had a helpful sort of application to get the user starting. We prepared a collection of tutorials, which are all didactic by intent. We had a certain amount of customization that enabled users to go and start looking at tracks. They could understand the nuances of working with Oxford Nanopore sequence data, and we're trying to go and appeal both to bioinformaticians who wanted to learn how we would solve particular problems using Nanopore sequence, and to sort of non-bioinformaticians saying, hey, it's not actually that scary. There are some sort of pretty preordained commands that you can use to go and analyze sequence data. So anyhow, it worked. The world was in order. Then, a couple of years ago, this nasty SARS-CoV-2 thing happened, and it perturbed all of our existences. One of the fundamental things that we found is we spun up a couple of tutorials to demonstrate how we could actually analyze SARS-CoV-2 sequence data in a Jupyter notebook. It was fabulous. It enabled users with Windows, with macOS, with Linux, to start understanding how we would use the Arctic protocols for analyzing the genomes. It did appeal to new bioinformaticians. It did appeal to already existing informaticians. It was a clean solution. But when we look at it as, is this really the way we should be analyzing data? It involves quite a lot of clicks. There are quite a lot of points where you, the user, needs to wait for a particular process to run. This is a horrible inefficiency. So we needed to have a little bit of a rethink, and how can we begin to implement workflows? How can we implement pipelines in our product so that we can actually empower a more tractable or a more meaningful bioinformatics analysis? So, that slightly scary figure on the right, that's just a lazy screenshot of a presentation that was given to one of the product management meetings, back in March 2021. This is when I outlined our plans. We are going to move to Nextflow. Why are we going to move to Nextflow? Well, hey, it's a DSL. It works. It's justifiable. There's NF core, so there's a little bit of a community around it. It kind of ticks off most of the things that we really need it to do, and it should be something that is magical just to install. A single apt-get, it should appear on the computer, and it should enable users to start running our workflows in a way where there's no clicking the button, there's no sort of waiting for things to happen. You fire off the command, data analyzes, you have results, and hey, it sounded like a really good plan. So we made the plan, this is what we're going to do. Of course, because we're appealing to biologists, because we're appealing to a particular cohort of users, we wanted to have rich reports. So we're coming up with our own best practices, which are parallel, but a little bit different to the NF core best practices, but which fit perhaps within the ecosystem that we're trying to develop and maintain. We like the explicit data flow. We like the scalability. We like the way that the platform could begin to satisfy our objectives and ambitions as the product develops. So let's have a look at a few of the workflows that we've developed. I'm just going to be talking about 3 workflows that exemplify the sort of challenges that we can resolve or we can work with using Nextflow. So on the screen behind us, we have what is called our metagenomic workflow, WF Metagenomics. Now, I don't need to tell anyone here anything about metagenomics, but for a lot of the applications that we've been looking at before, then there was this horrible dichotomy between what is amplicon-based metagenomics and what is whole genome metagenomics. If you have an application that works just on the 16S, then there's a vocal 15% of people who say, hey, why aren't you doing 18S? I don't want 18S, I want 28S and the ITS. So there's this horrible discussion as to what is ribosomal RNA analysis. So we thought, okay, let's come up with a workflow that can do everything that we would consider under true, shotgun metagenomics or through amplicon sequencing metagenomics. But the thing with metagenomics, if we ask why people are actually doing metagenomics, one of the key questions is often we want to know, is a particular organism within this sample or not? With nanopore sequencing, we have the data being available in real time, so we don't want to wait 20 hours or 40 hours for a sequencing run through which we can then begin the analysis. We want to start analyzing as soon as you've pressed go on the sequencing machine. You press go on the sequencing machine, and we expect the first FASTQ files to start appearing on the system within 3 or 4 minutes. This means that we can start producing actionable sort of data within the first, 3 or 4 minutes of a sequencing run starting. Do we really need to generate 10 gigabases of sequence if we're looking for a particular organism that we expect to be real well abundant within the sequence collection? So anyhow, this was a really nice example of how Nextflow didn't work. But this was also a beautiful example of how Nextflow does work. A couple of tickets, a little bit of heated discussion, a little bit of finger pointing, and then we had a solution. So thank you very much, Paolo. This was a ticket that was very gratefully received, and it's a wonderful demonstration that Nextflow will solve every problem that we are likely to fail. Don't get too comfortable. We've got a complaint slide coming in a few minutes. So, yeah, the world's great, but it's, there's always the but. Anyhow, metagenomics is a beautiful demonstration that we can do with Nextflow everything that we would like to be doing with Nextflow. Anyhow, let's look at some of the slightly more harder challenges. So everyone else has these beautiful, London Underground figures. I like those London Underground figures. I said to the team yesterday, having seen the posters, I want one of those figures for this presentation. And they said, but they're not automatically generated. Come on, Nextflow. Can't we have these figures produced automatically? This is, team, please. So, yeah, they're watching me. I'm not sure which camera it is, but yes, you're not forgiven. Anyhow, yeah, this is the usual execution plot that comes from Nextflow. And this shows the particular analysis that we're running here. Here we're taking a deep sequence single human genome. We've got long sequence reads, and what we want to do is we want to perform both the base calling and variant calling all in a single process. The base calling of Oxford Nanopore sequence data is fairly complex. It involves, machine learning. It involves neural networks. It involves a lot of GPU. So we've got the challenge here of how quickly can we turn around the analysis of a human genome. So here we can see that, we've got many hours spent just performing this base calling process. Why are we only using 8 GPUs? Well, discussion with some of the cloud colleagues here reveals that GPU is a very complicated commodity to go and get within the cloud. They are, rare, they are scarce. So we've got a challenge here that we've got a GPU limitation within the system. Hey, okay, that's fine, but the rest of the steps within the analysis parallelize extremely well. So we've got a turnaround time that allows us to deliver human genomes in a really sensible time. We've got a single-step workflow that goes from the raw data through to all of the VCF files and the bed files that we can then pass on to the tertiary analysis providers who can start saying, what is the actionable content? What is the meaningful content within this individual human genome? This is nice, this workflow, because we've got multiple sources of input. Are you starting with the primary data? Are you starting with unmapped data? Are you starting with mapped data? So we've got the vagaries of input here, which are all working towards a single standardized output. And then we've also got the beauty here that we can run it physically on device, the Promethean tower, the compute that comes with the system. That'll run natively, but it will also scale up to the cloud, or it can be run on your sort of cluster or physical hardware that you have available on site. So human variation, it's an important one, but it's just a demonstration of the complexity of sort of workflows that we're aiming to roll out to our customers. Perhaps there are some slightly easier ones. we've got a number of workflows that are looking at perhaps other slightly more defined data. So this one here is just a little bit of a pointer to a workflow that we have working on the understanding of what are potential antibiotic resistance genes within tuberculosis genomes. All we should be looking here is at the beautiful picture. We're working towards tractable, we're aiming towards reports that are informative, they're lightweight, and they can easily be distributed as an integral part of the analysis. So, tuberculosis, it is but one of many pathogens that we need to be considering as mankind moves forwards. So anyhow, that's Nextflow. It's super exciting. We have a problem, though. The problem is that we're trying to democratize bioinformatics. And we, as bioinformaticians, look at the console and say, yep, that's reassuring. It's what we like. It's what makes sense to us. Our colleagues in technical support, though, find that, encouraging non-bioinformaticians to use the console is heresy. It generates a lot of anxiety in the user. What do I type? Where do I type? What does it mean? What's Nextflow? Why do I have to spell it this way? These are sensible questions that, we as bioinformaticians, we just understand the answer, but as a non-bioinformatician, working at the console is a little bit mysterious. It is a little bit scary. It is a little bit challenging, and it really is a barrier to the usage of our Nextflow workflows. So what is our solution to this? Well, we have our Epitome Labs product, and what we're trying to do here is we're trying to go and provide a friendly desktop interface through which a non-bioinformatician can have the same experience. They run the workflow, but we provide them with the drop-downs. We provide them with the interface. We provide them with prepackaged workflows, and these are all available on the devices it ships, but they can also be installed by other customers onto their laptop as and when required. This means that we need to support running Nextflow workflows on Windows, macOS, and Linux. And this is something which, it's a little bit scary. We've got a Jupyter backend, so there's a whole lot of crazy logic in the workflow, but it's something which actually does seem to work, and we're getting very nice feedback from our user community. So how does a typical journey look like? So you're presented on your computer. Here's me on my Mac yesterday. And you're presented with a number of different workflows that you could be running. I'm going to say, hey, I'm interested in the Arctic workflow. This is our workflow for SARS-CoV-2 data, and it's something which runs in a sensible amount of time on a laptop computer in Barcelona. So you're presented with your drop-down, fairly simple dialogue, and I've got lots of traffic lights. If you look at where we would perhaps entered a name at the top, it says something like Nextflow Summit Demo. This was red before I typed it in. Each of our workflows would like a name. Then beneath, you can see there's another red icon. This icon's saying, hey, there's some information that the workflow would like. This is something which, you, the user, should absolutely be providing. So I don't have a screenshot of that, but anyhow, that was a file path. We needed to go and locate some data that was stored on my desktop. And then when everything's looking right, you're presented with a scary big green button. Well, it's not scary, a reassuring big green button. Hit the green button, and it starts off your workflow. Everything is working, and you're given a big button. Stop, I didn't put the right data in. I don't want my computer to sort of, start whirring and doing the things that it does. But anyhow, it starts accumulating all of the output that we require straight from the beginning. We're getting all of the logs from the running Nextflow process. We're a company. We have users who have expectations. We absolutely require this information because things go wrong. We want to understand which step in the process has gone wrong, so we're making it easy for our users to capture all of the information so that they can send it back to our technical support colleagues who can raise the tickets and can help us move on and solve bugs or add new features, depending upon the source of the error. Anyhow, we wait a few minutes, and we get to the point where actually it is finished. It's finished here to the extent that it's providing me with a collection of files, and it's got a nice button, Open Report. Hit the Open Report button, and here we have the SARS-CoV-2 report. Here we've just got an excerpt of the report. It's a fairly long HTML file, but within this file here we have the particular coverage plot that is giving us a little bit of insight as to what is the depth of coverage across the individual amplicons, and this is one of the key QC features that our users should be looking at before they then look at the strain identification and make the call that yes, this genome could or should be submitted to GISAID. So anyhow, Epitome Labs has been out for a few months now. This is a way that we can begin to provide Nanopore curated workflows that can be run on device or in a sort of on your laptop, on a cluster, more or less anywhere where you have access to a sort of a terminal and a little bit of screen. What about the future though? What should we be doing? So one of the challenges we have at the moment is we started out with Jupyter, and Jupyter is great, but perhaps it's not the best environment for high throughput data analysis. So we've said, okay, the next product is going to be entirely free of Jupyter. We're moving to something which is a little bit more Electron-like. So this is just a little bit of a glance as to what is coming in one of our next releases. So certainly in the next month or so, this will be hitting the wild. Here you can see there's a smaller number of workflows, but at the bottom right, there is a nice button, install a workflow. So let's see what happens if I install a workflow. Well, here I'm installing a workflow from NF Core. This means that instead of restricting our users to a subset of workflows that we have preordained as being suitable to run on our device, here we're saying, hey, why shouldn't we support any NF Core compliant type workflow? We're going to use the NF Core schema to work out what is it that we need to ask the user for in terms of information, but this means that our users will have access to a broader set of tools, and this means that users who want to develop tools for Nanopore users now have a clear path that working with the NF Core project or developing a workflow that is NF Core compliant will at least hopefully work on our device and undoubtedly help create a few more tickets. Anyhow, does it actually work? Let's hit import. Well, it thinks for a few seconds, it does a little bit of a git pull, and there we are. We've got an NF Core workflow that is in a position that we can run on the desktop. I was considering doing a live demo, but at the moment, who knows what could possibly go wrong. So no, anyone who would like a demo, do catch up with me afterwards, and I will convince you that this is real and it is possible. Anyhow, everything at the moment is the run-up to the ASHG conference in a couple of weeks' time, or it's just over a week's time. So it's human genomes at the moment. Let's see what we can do with human genomes in the new version of Epitome Labs. Well, we can open a screen. We can update our repository. We can delete our repository. I certainly can't run a human genome analysis on this laptop, but we can certainly have a look at a couple of simpler examples a little bit later on if you are interested. So please watch this space. We have a good idea as to a product that will hopefully be a little bit more applicable to other Nanopore users, and hopefully perhaps generate a little bit of development within the community. So anyhow, that's the good, that's the great, and that's the excellent. What are the challenges? So as a company, it's easy for me as a product manager to release workflows, to release code. It's our technical support team who pick up the problems, and they generate all sorts of tickets for us. The number one ticket that is perturbing very many of our users is the nonsense of, the junction between Nextflow and Java. This is particularly acute for users on macOS. If you often install a standard version of macOS in your computer, it's incompatible with the latest versions of Nextflow. There's just anxiety amongst the users. Anxiety amongst users generates tickets, and generate tickets, it takes time. We would like to have a simpler solution here. Could we imagine perhaps a world where we have a Nextflow that is distributed with its own JVM so that it just works? There isn't the requirement for dependencies. Another thing that we hear fairly often is other workflow engines. There's something a bit snaky. They have a concept of until. And this is where instead of running the whole workflow, you run to a particular point within the workflow. A load of you can say, hey, that's actually daft. Why would you want to run only part of the workflow? Well, sometimes you only want to create a subset of the files that you need so that you can start developing other bits. If you want to do a didactic deep dive into what a workflow is doing, you might want to look at particular points within the workflow and the results that have been generated rather than going right to the end. So, hey, perhaps there's something that could be considered there in the future. The biggest bugbear, however, is unexpected input curly bracket. I mean, come on. This is not really a dreadfully usable error message. The number of times that we get tickets, hey, we've got this unexpected input. What does it mean? Well, it means, okay, you need to send us a load of logs. You need to give us a little bit of proof that your input files really were what your input files were. It's not a nice conversation, and it's something which is very, very difficult for us to support because we need to have so much additional information. So anyhow, anything that could be done to give a little bit of additional sanity checking to the outputs that come from Nextflow going wrong would be gratefully received by our technical support team and, of course, our own development team who can better start understanding and handling these exceptions. So that's kind of me. Quick summary. We've got a product that works on most platforms. We quite like our platform. It's working and it's delivering to our users, to our non-bioinformatics users, a way that we can run workflows without having to sort of, really dive into the terminal and without having to face the struggle of installing a whole lot of software. The workflows are all run using Nextflow, and we're content with Nextflow. It's giving us a good experience, and we're happy with what we have. The fact that we've been able to implement real-time workflows is something which I feel is extremely important, and it opens up a number of exciting opportunities as to the sort of work that we could be doing and exploring in the future. We're going to be enabling generalized import from GitHub in the immediate future, and this should enable you to run any NF core-like workflow. We are a company. We do like to put our license and our terms and conditions on a lot of our software, but at the moment, all of the Epitome Labs software is entirely licensed under the Mozilla 2 public license. So at the moment, there is absolutely no hurdle to you using our workflows, to you using our interface, to exploring what can be done here. So, really, I'm super eager to see how we can move the community in different directions. And with that, I would say thank you very much.
Great, thank you very much, Stephen. We've got a couple of questions there. One of them is around, you mentioned to the last minute around the workflows being open source there. How adaptable are they for other species?
Yeah, good question. So we often give our workflows complicated names like human. Does human really imply that the workflow only works with human? It means that really we've only developed it for human. Yes, you have to provide a reference genome. So this means that WF human variation would also work as WF sort of chimpanzee variation or sheep variation. We're not bound to a single genome. Of course, it's not tested, and we can't imagine what could have gone wrong.
And then with regards to the compute, where is that running in the examples you showed us there? Does it connect into the user's own cloud environment, or are you running that within your own environment?
So all of the examples presented here all run on local resource. So we're not running these workflows in the cloud. That might be a conversation for next year.
Okay. And then where do you kind of see this on the sequencer compute going over the next years? I mean, we saw a couple of weeks ago with Illumina's new machines that's got a lot of compute in there. Is that some sort of direction that you're thinking of going as well?
Yes, of course. I mean, this is of course why we are developing these workflows. A lot of the sequencing devices have plenty of compute. There will be cases where users want to compute on device. There'll be other cases where they want to compute in the cloud. So we need a solution where you can compute wherever. Awesome.
Is there support in MinION for, is it MinION? MinION? That's a new one for me. To automatically trigger an Nextflow pipeline when a run is completed?
So internally, inside Minnow, there is an opportunity for Nanopore to run Nextflow workflows. This is not something which is available to the community, and it's not something which is supported yet.
Awesome.
Watch this space.
And there's a final request from Harshal is, can we use Guppy containers in NF core pipelines? Please, I think, pretty please, maybe.
So thanks, Harshal, for that question. Let's have a chat when we're not being recorded.
Awesome. Great. Thank you very much, Stephen.